Hi everyone, today's topic is Nucleosome Model, an interesting topic we studied in genetics. If you want to know how DNA is packaged in a chromosome, then you must need to study nucleosome or solenoid model for DNA packaging. In this video, we are going to discuss some basic concept of chromosome like what is chromosome, the concept of chromatin, what is nucleosome and its structure, and how DNA is packaged within a chromosome, and solenoid model. So what is chromosome? Chromosomes are thread-like compact structure of nucleic acid and proteins that readily fits inside of the nucleus. Chromosome present in both prokaryotes and eukaryotes. In the figure below, the stages of cell division is showing where in the prophage stage the chromatin fibers is ready to condense and start condensing to form a compact structure chromosome. So finally in the metaphase stage of cell division, it's showing the condensed and tightly coiled chromosome structure that is ready for further process, and then anaphase and telophase of cell division occurs gradually. Now we will discuss the concept of chromatin. So what is chromatin? This basic question always confuses me between the DNA strand and chromatin fiber. Now I want to make it clear that chromatin is a less condensed state of chromosome and loosely appear or present in a nucleus, while the cell is in interphase stage. Chromatin fiber is a mixture of DNA and proteins present in a nucleus as an indistinguishable mass or network that is tightly coiled and condensed to form a chromosome. In a word, chromatins are the unwound structure of chromosome. In the figure, we can see that how chromatin present inside of a nucleus. Next, we are going to learn what is nucleosome. Nucleosome is the basic repeating subunit of chromatin packaged inside of a nucleus in a manner of supercoiling. When a DNA strand is wrapped around some histone proteins, that leads to a formation of a bead-like structure called nucleosome. Nucleosome is 11 nanometer in diameter. The beads are interconnected by the DNA strand called linker DNA, and the total structure of DNA strand with both core DNA, which is wrapped around histone protein, and linker DNA looks like beads on a string. In the figure, it's showing an electron micrograph of chromatin isolated from interphase stage of cell division, where we can see a series of ellipsoidal beads in black are joined by thin and less dark colored threads. That's called beads on a string. So what are the components nucleosome made up of? Nucleosome made up of DNA strand and histone proteins, where 8 histone proteins create an octamer and the DNA strand coiled around the core histone octamer by 1.65 turn. And the histone proteins like such as 2 of each H2A, H2B, H3 and H4, and another histone protein H1 present as a linker with the DNA and histone octamer. In this figure, we can see that the core DNA wrapped around the histone proteins and the overall diameter of each nucleosome is 11 nanometer, and the core DNA is about 146 base pair in length. The complete nucleosome is 166 base pair that form almost 2 superhelical turns of DNA around the histone proteins, and H1 stabilize the nucleosome conformation. Let's get to know how DNA is packaged within a chromosome. Generally, eukaryotic chromosome contain nearly 2 meters of DNA in length. During cell division, in case of both mitosis and meiosis, the DNA need to be coiled to be fit inside of a nucleus, because a DNA molecule can't proceed its function in a linear form easily, and also the linear DNA is easily accessible for any enzymes like degrading enzymes and not stable inside the cell. That's why DNA packaging is important, because it protects the DNA from damage, can be transmitted easily during cell division. Thus the chromosome provides overall stability of a DNA. The DNA must be compacted in many orders of magnitude to be fit inside of a small size nucleus, and the DNA molecule somehow tightly coiled and densely packed into the compact metaphase chromosome structure. The condensation occurs by 10 to the power 4 fold from the linear state of DNA molecule. Here the figure is illustrated for understanding the nucleosome model. Having 3 levels of DNA packaging are at the first level of DNA packaging, 2 nanometer linear DNA start coiling with nucleosome core particles and make it 11 nanometer in diameter. and overall structure is called bits on a string. And the second level of DNA packaging involves the condensation of the 11 nanometer nucleosome chromatin fiber to form more condensed and 30 nanometer supercoiled structure known as solenoid. And the 30 nanometer solenoid chromatin structure is stabilized by histone linker proteins and some other non-histone chromosomal proteins. Then the chromatin fiber coiled more and form 300 nanometer, then 700 nanometer to form scaffold structure. And then third level of DNA packaging, the giant DNA molecule supercoiled and condensed more to form 1400 nanometer chromosome, that is 10 micrometer long, is called metaphase chromosome, and the total DNA packaging is called nucleosome model of DNA packaging. Thanks for watching. Subscribe for more.